Then today we will start with the net gain of glycolysis. What are the things, what are the outcome as far as the glycolytic process is there? So net gain of glycolysis. Now actually at the end of glycolysis, we know there are two molecules of 3-carbon pyruvic acids are formed from one molecule of glucose containing 6 carbon. So that is the first obtained product, that from one glucose molecule containing 6 carbon, two molecules containing 3 carbon pyruvic acid, they are produced. Secondly, only 2 ATP are utilized, and that 2 ATPs are utilized for what? For phosphorylation of glucose to glucose 6 phosphate and fructose 1,6 diphosphate. There 2 ATP molecules are utilized, and NADPH2, they are produced. 2 NADP in all they are produced. And during ETS, towards the end of the chapter, we will see that electron transport system, from 1 NADPH2, 3 ATPs are produced. So here in glycolysis, 2 NADPH2 means what? 6 ATP molecules will be produced. Then during substrate level phosphorylation, means where addition of inorganic phosphate directly takes place without involvement of organic phosphate, and as such during that substrate level phosphorylation, 4 ATPs are directly synthesized, they are produced. And as a whole, if we see net gain, 8 ATP molecule will be produced, 4 from the substrate level phosphorylation, 4, and 6 from the NADPH2, means total 10. And out of that 10, 2 are utilized for phosphorylation of glucose and glucose 6 phosphate. That 2 ATP will be minused, they are used. So net gain, there will be 8 ATP molecules. So we can say at the end of glycolysis, we can obtain 8 ATP molecules. Actually, they are 10, but 2 are used. So it will be 8 ATP net. And the general chemical equation, as far as glycolysis is concerned, you can see C6H12O6, that is nothing but the glucose, right? And 2 ADP are used. And as 2 ADPs are used, they will be converted to ATP, and 2 NADPH2 are produced from 2 NAD. So the general equation, C6H12O6 glucose, 2 ADP are required, 2 NAD are required, and they are converted to 2 molecules of C3H4O3. What is that? It is nothing but the pyruvic acid, 2 molecules, and 2 ATP along with the 2 NADH2, they will be produced. Now here you can see the exact utilization of ATP. Number 1, पहिला ATP ग्लुकोज टू ग्लुकोज 6 फॉस्फेट दिसतोय. दूसरा ATP जो आहे तो फ्रुक्टोज 6 फॉस्फेट टू फ्रुक्टोज 1,6 डायफॉस्फेट, 2 ATP वापरलेले दिसतायत का?
हो सर.
Okay, then see where the ATPs are produced. Firstly, it is during 1,3 biphosphoglycerate to 3-phosphoglycerate. दिसतंय का? 1,3 biphosphoglycerate to 3-phosphoglycerate, 2 ATP will be produced. And lastly, phosphoenol pyruvate, it is converted to pyruvic acid. Last reaction, again 2 ATP will be produced. So 2 plus 2, 4 direct ATPs are there. And in addition to that, you can see there is a synthesis of NADPH2, where it is, here you can see NADPH2 will be produced from glyceraldehyde 3-phosphate to what we call it as an 1,3-diphosphoglycerate. And as a result of that, 2 NADPH2 will be produced. From 1 NADPH2, 3. So 3 into 2, 6 plus 4. So total 10 ATP are produced, but don't vaporlate already. So it will be 8 ATP molecules which are there. Are you getting this? Second process, it is nothing but oxidative decarboxylation of pyruvic acid. It is called as oxidative decarboxylation of pyruvic acid, which is actually produced during the process of glycolysis. Am I right? Now, this process is actually decarboxylation of pyruvic acid. Means what? Pyruvic acid contain how many carbons? Can anybody answer? Yes or no? Yes or no?
No, sir.
अरे गाढ़वा, पायरुविक एसिड कंटेन 3 कार्बन्स. कितना कार्बन रहता है? 3 कार्बन्स, right? And in mug, if we see the actual process where you can have this one, okay, can you able to see the screen?
Yes, sir.
दिसते ना?
Sir.
Okay. तुम्ही कावर दिसतंय सगळे मला? Yes. Here you can see. Yes. It is, we can say 3 carbon. दिसते ना स्क्रीन? दिसते ना? काय प्रॉब्लेम नाही ना?
नाही सर.
हो
Okay.
सर,
sir.
दिसते.
दिसतंय, काय प्रॉब्लेम नाही. Now, actually, if we see the pyruvic acid which is containing 3 carbon, आता या 3 कार्बन pyruvic acid मधनं decarboxylation means what? It will be the carboxylation and decarboxylation. We actually call it as an decarboxylation means removal of carbon dioxide. मग 3 कार्बनच्या pyruvic acid मधनं एक CO2 काढला, किती कार्बनचं कंपाऊंड उरेल? जर 3 कार्बनच्या कंपाऊंड एक कार्बन डायऑक्साइड काढून टाकला तर किती कार्बन उरतील?
2.
ओनली 2 कार्बन विल बी देअर. आणि 2 कार्बनचं जे कंपाऊंड असतं त्याला आपण म्हणतो ऍसिटिल. 2 कार्बन ऍसिटिल. सो देर विल बी फॉर्मेशन ऑफ ऍसिटिल formation. Acetyl moiety will be formed, and that's why it is also called as acetylation process. Let's see what does it mean. As this, it is a step 1 in the respiration during which carbon dioxide is removed. It will be the first decarboxylation that occur here, because during glycolysis there wasn't any removal of carbon dioxide. पहिल्यांदा carbon dioxide बाहेर पडणार आहे. So it will be the first decarboxylation that occur. Secondly, in this step, pyruvic acid which is generated in the cytoplasm by the process of glycolysis, it enters into mitochondria. A very interesting fact is that during glycolysis it was pyruvic acid was produced. ते सगळं cytoplasm मध्ये होतं. But from this pyruvic acid it has to enter into the mitochondria. But mitochondrial channel proteins are small. त्याच्यातनं 3 कार्बनचं कंपाऊंड जाऊ नाही शकत. मग इट इज ट्रिम्ड, इट इज कट, आणि एक कार्बन डायऑक्साइड काढून टाकला की देर विल बी फॉर्मेशन ऑफ what we call it as an 2 कार्बनचा acetyl moiety. आणि 2 कार्बनचा acetyl moiety it can enter into mitochondria. So we can say in this step pyruvic acid generated, produced in the cytoplasm at the end of glycolysis, it can enter into mitochondria in presence of a enzyme, pyruvate dehydrogenase enzyme complex. Pyruvate dehydrogenase, pyruvic acid will undergo dehydrogenation. The enzyme is there, and NAD plus pyruvic acid, pyruvate undergo oxidative decarboxylation. 2 processes at a time. Oxidation means removal of hydrogen, and decarboxylation means removal of carbon dioxide. As a result of that, 2 carbon compound will be produced, moiety will be produced. It is called as acetyl moiety. Now that acetyl moiety will combine with coenzyme A at the outer mitochondrial membrane. At the outer mitochondrial membrane, acetyl moiety plus coenzyme A, they combine together to form acetyl-CoA. Acetyl-CoA will come to the inner membrane of mitochondria and it will relieve the acetyl moiety inside the mitochondrial matrix for further processing. During this process, NADPH2 will be produced. Obviously, that hydrogen will be accepted by NAD and it will be converted to NADH2 and CO2 will be produced. Then this formation of acetyl moiety actually takes place at the outer membrane. of the mitochondria that is in the cytoplasm, and then it will come across inner membrane and then relieve the acetyl moiety. So it will be completed in the mitochondria. This conversion of 3-carbon pyruvic acid into 2-carbon acetyl-CoA, it is called as acetylation or it is called as link reaction. त्यालाच आपण काय म्हणतो? Link reaction. And then if we see the next step, wait a minute. Share button कुठे गेलं? Yes. Okay. So that is the step 1. And then the next step we can discuss. It is nothing but the acetyl-CoA. It enters into the Krebs cycle for further degradation. And mug, एकदा का acetyl moiety comes into the mitochondrial matrix. पुढची step, third step, Krebs cycle सुरुवात होणार. Now that acetyl-CoA, it is called as connecting link between glycolysis, that is cytoplasm, and Krebs cycle, that is mitochondrial matrix. मग outside the mitochondria, glycolysis. Inside the mitochondria, Krebs cycle. या दोघांना जोडण्याचं काम, कनेक्ट करण्याचं काम कोण करतं? Acetyl-CoA, and that's why it is called as connecting link. That acetyl-CoA is substrate entrant into Krebs cycle, where the Krebs cycle will start with the receiving acetyl-CoA. During the oxidation of pyruvic acid, hydrogen is removed. This hydrogen is accepted by NAD and it will be converted to NADH2. Then 2 molecules of NADH2 are formed from 1 glucose, that is 2 pyruvic acid molecule, which produces how many ATPs? 1 NADPH2 3, means totally 6 ATP will be produced during the terminal oxidation. So the overall, we can say reaction for link reaction, it is pyruvate, pyruvic acid plus NAD plus coenzyme A plus water molecule for hydrolysis. So pyruvate dehydrogenase enzyme complex will be there, and there is a formation of acetyl-CoA plus NADPH2 plus CO2. So acetyl-CoA will be end product, and that acetyl-CoA will enter into what? Krebs cycle, or it is called as TCA cycle, or it is called as citric acid cycle. Now, why it is called as Krebs cycle? Because Sir Hans Krebs, 1937, discovered this cycle, and that is in pigeon muscle. It is called as TCA, tricarboxylic acid cycle. All the organic acid will contain 3 carboxylic acid group. That's why it is called as TCA cycle. Then the first stable compound during this cycle, it will be citric acid, right? And as it is a citric acid, which is a stable compound, we can call it as an citric acid cycle. Are you getting this? Why it is called as Krebs cycle? Because Sir Hans Krebs discovered this cycle, and that's why it is called as Krebs cycle. TCA cycle, all the organic acid will contain 3 carboxylic acid group, COOH group. That's why it is called as tricarboxylic acid cycle. Then citric acid cycle, the first stable compound is citric acid, and that's why it is called as citric acid cycle. Now, it occurs in the mitochondrial matrix, and that too in presence of free molecular oxygen. 2 things. Number 1, it will take place within mitochondria matrix, and number 2, free molecular oxygen, it is available. Then it is representing the third step in the aerobic respiration, not second, it is third. The first one is glycolysis, second one is acetylation, and this is the third step. In this cycle, pyruvic acid, it is further undergoing degradation to form complete oxidation product CO2H2O. Complete oxidation of pyruvic acid will result into formation of CO2 and H2O. That's why it is called as complete oxidation. As I have told you, it was first discovered by Sir Hans Krebs, 1937. And as he discovered that in a pigeon muscle, he awarded with Nobel Prize in 1953. It was pigeon muscle where Krebs cycle was discovered. For neat purpose, you have to remember that. As the citric acid is the first stable compound of this cycle, it is also called as citric acid cycle, right? Similarly, various organic acid involved in the cycle, they contain 3-COOH group, carboxylic acid group. Therefore, it is known as tricarboxylic acid cycle, or simply it is TCA cycle. And each and every step of the Krebs cycle, it is controlled and catalyzed by specific enzymes. And then we'll go for the actual Krebs cycle. Here you can have the Krebs cycle, as well as along with that, there will be the appearance of what we call it as the acetylation process. Now, can you able to see?
Yes, sir.
Now, here
Yes,
you can
sir.
see carefully. Here you can see this is the process that is called as acetylation. Here you can see during glycolysis, glucose 6 carbon undergo glycolysis to produce 2 molecules of pyruvic acid containing 3 carbon. Now at the outer membrane of the mitochondria, this pyruvic acid undergo oxidative decarboxylation. Oxidation means removal of hydrogen. They will be accepted by NAD. NAD will be converted to NADH2. Then 1 molecule of CO2 will be directly removed and it will be given out. And as a result of that, 2 carbon moiety will be produced, acetyl moiety. This acetyl moiety will combine with coenzyme A, and that acetyl moiety with coenzyme A, collectively it will form acetyl-CoA. Now, this acetyl-CoA can enter into mitochondrial matrix where it can undergo the process of Krebs cycle. Are you getting this? Yes or no?
Yes, sir.
Sir.
Yes.
Okay. And then if we see the acetyl-CoA that releases acetyl moiety, it combines with 4 carbon oxaloacetic acid. The 4 carbon oxaloacetic acid will combine with, here you can see 4 carbon oxaloacetic acid. It combined with acetyl moiety containing how many carbon? 2, right? And there will be the involvement of water, that is hydration. As a result of that, 4 carbon acetyl moiety, 4 carbon oxaloacetic acid plus 2 carbon acetyl moiety. Collectively, it will form citric acid containing how many carbon? 6. So that is the first stable compound, citric acid, produced by condensation of acetyl moiety and 4 carbon oxaloacetate. Now this 6 carbon citric acid, it undergo dehydration, removal of water molecule, and there will be formation of cis-aconitic acid, or it is called as cis-aconate. Now this 6 carbon 6-aconate undergo hydration. पहिल्यांदा डिहायड्रेशन झालं होतं, आता हायड्रेशन होतं, and there will be formation of isocitric acid, again containing 6 carbon. Now this isocitric acid, it undergo oxidation. The hydrogen will be removed, it will be accepted by NAD, and NAD will be converted to NADH2 along with the formation of oxalosuccinate, again containing 6 carbon. Are you getting this? The citric acid undergo dehydration, cis-aconitic acid. Cis-aconitic acid undergo hydration to form isocitric acid, and then that isocitric acid will be undergoing oxidation to remove hydrogen, that will be accepted by NAD to form NADH2, and there will be formation of 6 carbon oxalosuccinate. Now that oxalosuccinate containing 6 carbon, it undergo decarboxylation. Now here you can see there is a removal of carbon dioxide. Out of 6, 1 carbon dioxide removed, so it will remain 5 carbon. Now that 5 carbon compound is called as alpha ketoglutarate or alpha ketoglutaric acid, alpha KGA, containing 5 carbon. Now this 5 carbon alpha KGA again undergo oxidative decarboxylation. During oxidative reaction, NADH2 will be produced, and during decarboxylation again 1 CO2 will be released. So that will be the second CO2 which is released. So 4 carbon alpha KGA, alpha ketoglutaric acid, undergo oxidative decarboxylation. During decarboxylation, CO2 will be removed. During oxidation, hydrogen will be added to NAD, which is converted to NADH2. As a result of that, 4 carbon moiety will be produced. That is called as succinyl moiety. Succinyl moiety again combined with coenzyme A, and there is a formation of succinyl CoA. Now this succinyl CoA undergo hydration, or you can say hydrolysis, and during that hydrolysis 1 ADP will be produced, 1 ADP will be converted to 1 ATP. The actual reaction is 1 GDP, guanosine diphosphate, will be converted to guanosine triphosphate, but they are equivalent to ATP. So we can directly say it is the ATP which is produced, and a 4 carbon organic acid will be produced, and that is called as succinic acid. Now this succinic acid again undergo oxidation, but this time no NAD. Instead of NAD, there will be flavine adenine dinucleotide, usko bolte hain FAD. That FAD will receive the hydrogen from succinic acid, and it will be converted to reduced coenzyme FADH2, flavine adenine dinucleotide. It is reduced by using the hydrogen from the succinate, and then it will be converted to a 4 carbon fumaric acid or fumarate. Now this fumarate, once again, it will undergo hydrolysis, and it will be converted to another organic acid that is called as malic acid. Now this malic acid is again undergoing oxidation. NAD will be used, NADPH2 will be produced, and that malic acid will be converted to oxaloacetic acid, which is available for repeating the process. So oxaloacetic acid will be again regenerated. It will available for repeating all the processes. So that is how a 3 carbon pyruvic acid by link reaction enter into the Krebs cycle. In the Krebs cycle, that acetyl moiety containing 2 carbon, it will undergo decarboxylation, and during decarboxylation, the first carbon will be released during oxalosuccinate to alpha ketoglutarate, and from alpha ketoglutarate to succinyl CoA. So there will be total loss. There will be total loss of what we call it as an carbons which are present in the pyruvic acid, or you can say it will be what we call it as an acetyl moiety, right? Yes. It will be the net gain of ATP during Krebs cycle. How many ATPs are generated during Krebs cycle? Already we know 6 NADPH2 are produced. So 6 into 3, it will be 18 ATPs. Then 2 FADH2 are produced. They are converted to 2 ATPs. So 2 into 2, 4 ATPs. And 2 GTP are produced. They are equivalent to 2 ATPs. So net gain total 24 ATP are produced from 2 molecules of pyruvic acid. Obviously, it will require the information that 1 molecule of NADPH2 will produce 3 ATP. 1 molecule of FADH2 will produce 2 ATP, and 1 molecule of GTP is equivalent to 1 molecule of what? ATP. So that will be the total gain, total availability of ATP for glycolysis and followed by Krebs cycle. It will be 24 ATPs. So this is the